Okay, so let's move on to the cytoplasm. The cytoplasm of the cell is the material outside the nucleus and inside of the plasma membrane. The cytoplasm appears clear with scattered specks underneath the microscope. The clear substance is known as cytosol and is jelly-like in consistency. Cytosol provides the fluid medium necessary for biochemical reactions. In addition to cytosol, the cytoplasm also contains something called inclusions, which are temporary chemicals and nutrients such as glycogen, lipids, and pigments. These do not function and do anything. They're just kind of there. Cytoplasm also houses the cell's various organelles, which are the membrane-enclosed bodies in each cell. Each organelle is charged with a specific function. Just as body organs work together to keep the body functioning, the organelles work together to keep the cells functioning. The primary organelles within a cell are the ribosomes, the endoplasmic reticulum, and there is a smooth and a rough one, the Golgi apparatus, the mitochondria, the lysosomes, the peroxisomes, the cytoskeleton, and the centrioles. And we're going to talk about each one individually and what they do. So as you may recall, the ribosome is an organelle that serves as the site of protein synthesis. It is made up of two rRNA subunits, and they're wrapped around an mRNA unit, and they start translation. Unlike most other organelles, ribosomes are not contained within a membrane, and therefore they kind of free float all within the cytoplasm and adhere to the rough endoplasmic reticulum, another organelle we'll talk about in a little bit. The endoplasmic reticulum exists in two forms, the rough ER and the smooth ER. The rough ER is dotted with those ribosomes, giving it a bumpy appearance. The rough ER is tasked with synthesis and modification of proteins that are designed for the plasma membrane. A ribosome on the rough ER synthesizes or makes a protein. That protein is then released into the channel of the rough ER, where it is bonded with sugar and transported to a vesicle to be packaged and shipped in the Golgi apparatus. A vesicle is just this little thing that carries things around. The smooth ER lacks all those ribosomes, and they're responsible for the synthesis of lipids. Lipids are also known as fats. The smooth ER synthesizes phospholipids and steroid hormones, so they make those. Cells that produce large amounts of hormones, such as those in the ovaries, contain large smooth ERs. The smooth ER also stores and regulates calcium, metabolizes carbohydrates, and breaks down toxins. The Golgi apparatus is responsible for sorting, modifying, and shipping off products that come from the ER. The Golgi apparatus looks like a stack of pancakes. On one side of the stack, the apparatus receives products from those vesicles. These products are then sorted through, repackaged on the other side, and are sent out of the cell. Most products are sent via vesicles that exit the cell. Some products are sent to the cell membrane to assist in structure, and some become lysosomes. Now, lysosomes are sent out by the Golgi apparatus to break down certain materials. Lysosomes contain enzymes that break down and digest unneeded cellular components. Some items they digest may include damaged organelles, foreign material that enters the cell, and sometimes they'll even eat themselves. Autophagy, which means self-eating, is the process of the cell digesting its own structures. In circumstances when a cell becomes unhealthy, they'll self-destruct in a mechanism called autolysis. Autolysis is a controlled cell death. The cell death within a cell cycle is known as apoptosis. Now, mitochondria are bean-shaped organelles responsible for energy production. The inside of a mitochondria is lined with winding structure called cristae. Along this inner membrane is a series of proteins, enzymes, and molecules that come together to produce biochemical reactions, leading to cellular respiration. These reactions convert energy from stored nutrients, such as glucose, into adenosine triphosphate, which is ATP. ATP provides a usable cellular energy to the cell. Certain areas that constantly use energy, such as the muscles and nerves, have a large number of mitochondria. Peroxisomes are organelles made up of mostly enzymes. They are responsible for lipid metabolism and chemical detoxification. Unlike the digestive enzymes and lysosomes, the enzymes in peroxisomes transfer hydrogen atoms to oxygen, producing hydrogen peroxide. This is what peroxisomes use to neutralize poisons such as alcohol. Now, just like the skeleton supports the structure of the human body, the cytoskeleton supports the structural integrity of a cell. The cytoskeleton is a group of fibrous proteins that extend throughout the cytoplasm. These proteins include these three types of filaments: a microfilament, an intermediate filament, and a microtubule. The thickest of the three are microtubules. They hold cell shape and resist the compression of the cell. The microfilament is a thinner cytoskeleton filament that is composed of actin. Actin is a protein that, when twisted together in chains, are responsible for muscle contraction, and they also are responsible for splitting the cell during cell division. Intermediate filaments, which are the middle-sized filament, is made up of keratin and is wound together like a rope. While the microtubules resist compression, the intermediate filament resists tension and keeps forces from pulling cells apart. Some cells have cell projections. These projections are appendages made up from microtubules. Cilia are found on some cells, such as the epithelial cells that line the respiratory tract. The cilia moves rhythmically, and they beat constantly, moving waste such as dust or mucus upward away from the lungs. A flagellum is a longer appendage that is specialized to move the cell. The only cell in the human body with a flagella is a sperm cell, which needs to be able to propel itself towards a female egg. Another microtubule structure is called a centrole. These are located near the nucleus and serve as the origin point for cilia or flagella. Centroles can also assist in separating DNA during cell division. Now that we've discussed the anatomy of a cell, we're going to start discussing the physiology of a cell. One of the great wonders of the cell is the cell's ability to move substances in and out of the cell through the cell membrane. All substances that move through the membrane do so by one of two primary methods, each which are categorized by whether they use energy or not. Passive transport is the movement of substances across the membrane without the need of an energy expenditure. Active transport is the movement of substances across the membrane using energy, specifically ATP. To really understand the transport of substances across the membrane, there are concepts and terms you must be familiar with. A solution is a homogeneous mixture of two or more components. A solvent is a dissolving medium, like water, and the solute is the components within a solution that is smaller in quantity. Recall that the intracellular fluid is the fluid within the cell, and the interstitial fluid is the fluid on the exterior of the cell. Don't get interstitial fluid confused with extracellular fluid. Interstitial fluid surrounds the outside of a cell, essentially bathing it. Interstitial fluid is a component of extracellular fluid. Extracellular fluid denotes all body fluid outside the cell. The lipid bilayer structure of the cell membrane gives the cell its ability to be selectively permeable. The membrane allows substances meeting certain criteria to pass through, while others are repelled back by those hydrophobic tails. Diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. There are three main types of diffusion. Simple diffusion is where particles move through the membrane pores unassisted, while facilitated diffusion uses a protein to assist the particle with moving across. An example of facilitated diffusion is glucose. Glucose is too large to pass freely through the channels, so a glucose protein carrier transfers the molecule across. Osmosis only moves water freely across the cell, either through protein channels or by slipping between those lipid tails. A concentration gradient is the difference in concentration of a substance across a space. Molecules will spread or diffuse from where they are more concentrated to where they are less concentrated, until they are equally distributed in space. When a substance exists in greater concentration on one side of the cell membrane, the substance will move down its concentration gradient by crossing the cell membrane. Because this process does not require any energy to occur, it's considered passive transport. Now, during active transport, ATP is required to move a substance across a membrane. This often occurs with a protein carrier, and it's against the concentration gradient. There are two common forms of active transport. One is solute pumping, and the other is bulk transport. Now, in solute pumping, a protein serves as a pump to push a molecule against the concentration gradient. The most common solute pumping mechanism is the sodium-potassium pump found in many cells, but largely that pump is found in nerve cells. In the sodium-potassium pump, sodium is pushed out of a cell, causing potassium to move into the cell. Then there's the bulk transport method, which can be either endocytosis, which means bringing into the cell, or exocytosis, which means exiting the cell. Essentially, the cell ingests the molecule by enveloping it into a portion of the cell membrane. That portion pinches off and becomes a vesicle to move the molecule to its destination, either inside the cell or outside the cell. In endocytosis, depending on the consistency of the molecule, a cell may eat the substance by breaking it down and digesting it, or the cell may drink the substance by dissolving the fluid. The process of eating a molecule is phagocytosis. This occurs in the immune system, while the process of drinking a fluid is pineocytosis. A single turn of the cell cycle consists of two major periods. The first is interphase, followed by cell division. In interphase, the cell grows and carries out all normal functions and processes. Interphase is divided into three phases, the G1, S, and G2 phases. The G1, also known as the gap phase 1, is where the cell grows. In the S phase, or synthesis phase, the cell replicates its DNA. And during the G2 phase, the gap phase 2, the cell continues to grow and prepares for mitosis. Cell division consists of two separate phases, mitosis and cytokinesis. Mitosis is the general division of genetic material, during which the cell nucleus breaks down and forms two new fully functional nuclei. In cytokinesis, two distinct cells are divided from the cytoplasm. There are four distinct stages of mitosis. They are prophase, metaphase, anaphase, and telophase. I encourage you to read these definitions and remember them. There are some practice study guides inside your extra folder for you to look at. Just remember that prophase is the first part, metaphase comes in the middle, anaphase pulls apart, and telophase, you have two new cells.